welcome to my channel. Welcome to another video where we will be discussing biological classification. Okay, so this chapter is very important for your class 11. Okay, this is one of the base chapters. So first of all, we need to know what do you mean by classification. Okay, so the word classification means to classify or divide. Okay, suppose you have been given a task to form two teams. Okay, so how will you form two teams from your whole class? You will have few criterias, right? Suppose you need to form a football team. Okay, so what criteria you will see? Obviously, if you are selecting a goalkeeper, height is a criteria to select him. So the people with a good height will become the goalkeeper. Okay, so that is how we divide things. Another example is your teacher asked you to divide the class into two. So what you did is the people who got 80% above were placed in group A and who got less than 80% were placed in group B. This is how we classify. Okay, so in the first lecture we will be talking about Aristotle classification, 2 kingdom classification, 5 kingdom classification, 6 kingdom classification, and the kingdom Monera. Okay, so let's start discussing. Obviously, there are millions of organisms in the world, so it is very hard if we want to focus in a single species. It becomes very much difficult to know their properties. So what scientists did, they started a system of classifying them. The first classification was given by Aristotle, who was known as the father of biology. Okay, he distinguished between organisms by looking in their simple morphological characters. He classified living things as plants and animals. Okay, in case of plants, he divided the plants into herbs, trees, and shrubs. Obvious thing, the herbs were having a soft stem, the trees were having a woody stem. Okay, and a very fat bark. Okay, and the shrubs were having several woody stem but not as hard as the trees. Similarly, he classified animals on the basis of their RBC. The animals with RBC were kept in Anima and the animals without RBC were kept in Anima. The classification Aristotle gave was not that accurate. So, Carolus Linnaeus, who was considered as the father of systematic botany, gave another classification system in 1758, which was known as the 2 kingdom classification. It is important. Please remember, he wrote the book Species Plantarum and Systema Naturae. Species Plantarum referred to plants and Systema Naturae referred to animals. Okay. And the main basis of classification was the presence or absence of cell wall. Obvious thing, Carolus Linnaeus focused on only one thing. If there was a cell wall, the plants, the organisms which have a cell wall were kept in plants, and the organism which lacked a cell wall were kept in animals. Okay. So there were few demerits. That's why the two kingdom classification also failed. What were they? It was unable to distinguish between the unicellular and multicellular, the prokaryotic and eukaryotic, the photosynthetic and the non-photosynthetic. Okay. See, this was the system of two kingdom classification. Then came the most followed classification system, the five kingdom classification. Okay. So it was given by Robert H. Whitaker. He proposed five kingdom classification system in 1969. Remember the date, remember the year, then it was 1969. He established the kingdom fungi. Okay. So in Linnaeus system there was no fungi. There was only plants and animals, plantae and animalia. But fungi was introduced by R.H. Whitaker. Okay. And this classification system was based on the structure of cell, the organization of the body, the mode of nutrition, the reproduction and phylogenetic relationship. Okay. Now, the five kingdom classification, the kingdoms were Monera, Protista, Fungi, Plantae and Animalia. Okay. So we will be discussing in our lecture about all the kingdoms. Okay. Not in the first lecture but in the upcoming lectures. In the first lecture we will be discussing only about the kingdom Monera. Okay. Now see the five kingdom classification. Okay. So organisms were divided based on their cell type. Cell type means if they are prokaryotes, that is nucleus absent, either they are eukaryotes, that is nucleus is present. Okay. If they are eukaryotes, we look again into their body organization. They can be unicellular, that means a single cell. In case of eukaryotes also, they can be unicellular and multicellular. Okay. The example of unicellular prokaryotes is the Monera. Okay. The kingdom Monera comes over here. The example of eukaryotic unicellular organism comes the kingdom Protista. Then comes the cell wall. Okay. See, in the multicellular with cell wall are the kingdoms Fungi and Plantae. But fungi do not do photosynthesis, but Plantae do photosynthesis. Whereas Animalia has no cell wall. Okay. So like that it was continued. Again, next came The six kingdom classification system given by Carl Woos. Here he told that there were three domains and the classification was based on 16S rRNA. Okay, as 16S rRNA is a conserved sequence across all kingdoms. So the three domains were archaebacteria and eukarya. So the six kingdoms were archaebacteria, eubacteria, protista, plantae, fungi and animalia. Clear? So in the archaea comes the archaebacteria or the primitive bacteria. In the eubacteria comes the true bacteria. And in the eukarya comes protista, plantae, fungi and animalia, which are eukaryotic in nature. Okay. Until this, I think you all are understanding, right? Now comes the first kingdom. Okay. The kingdom Monera. All the bacteria are lying in this kingdom. Okay. The main followed classification system is the 5 kingdom classification. Okay. It is the most followed classification system. So, see, I have given an image where you can easily understand the size of a virus, the bacteria and an animal cell. Okay. So what are the characteristics? So the bacteria are prokaryotes, that is, they have no nucleus. Ubiquitous means found almost everywhere. They have double-stranded circular DNA. Suppose inside the bacteria, suppose this is a bacteria. So inside it there is its DNA. The DNA will be circular. Okay. Like this. The DNA will be like this. It is circular DNA. Okay. Membrane bound cell organelles are absent. There is no membrane bound cell organelles. The cell wall is very rigid or hard. The type of ribosome found in prokaryotes is 70S. Remember it. They can be motile. That is, this is the flagella of the bacteria. Suppose this is a bacteria and it is having a flagella. It will be able to move. The nutrition will be heterotropic or autotropic. Okay. Autotropic means can make their own food. Okay. And heterotropic means cannot make their own food. Okay. Now comes the classification of bacteria. Based on their shape, there is a the bacteria can be cocci, that is round shaped. Okay. The bacteria can be bacilli like, that is rod shaped. It can be spirilia like. Okay. That is thread like. And vibrio, that is comma like. Okay. So these are the shapes of a bacteria, classifying a bacteria basis of their shape. So now, what type of reproduction does a bacteria undergo? It can undergo both asexual and sexual reproduction. In sexual reproduction, we know fusion of gametes occur. In asexual reproduction, no fusion of gametes take place. Okay. So, asexual reproduction can be of two types, that is binary fission, where from a single parent C, single bacterial cell, by fission two bacteria are getting produced. Okay. And another is sporulation. Okay. So suppose this is a bacterial cell and this yellow color structure is known as the bacterial spore. Okay. These spores, when it gets outside the body of the bacteria, it can form a new bacteria. Okay. When the spore is inside the bacteria, sorry for the interruption, when the spore is inside the bacteria, that is known as the endospore, and when the spore is outside the bacteria, that is known as exospore. Okay. Now comes the conjugation. See, in conjugation what happens, two bacteria get attached with the help of a tube like structure, and genetic material, see this is the genetic material and it is getting transferred to another bacteria. Suppose this is one bacteria, this is another. That is known as the process of conjugation. Okay. So kingdom Monera is further divided into archibacteria, that is primitive bacteria, and eubacteria, that is true bacteria. Okay. In archibacteria there are halophiles, thermoacidophiles and methanogens. In eubacteria there is cyanobacteria, heterotrophic bacteria, actinomycetes and mycoplasma. Okay. So we will be discussing all this. Now in archibacteria they are the most primitive groups and they are present in the extreme of the extreme habitats. Okay. Their cell wall is made up of pseudopeptidoglycan. Pseudopeptidoglycan is a carbohydrate. Okay. Now comes the halophiles, the thermoacidophiles and methanogens. Okay. So the halophiles are present in the saline environments. The example is halobacteria. So halophiles are present in saline environments where there is extreme saline or salty waters. It is seen in the Great Lake of USA. Thermoacidophiles are present in high temperature and low pH. Okay. Sulfolobus is an example. And methanogenous bacteria are present in gut of ruminants. Okay. Example is methanobacterium. It is present in the gut of cow, buffalo, organisms like that. Clear? Now comes the eubacteria or the true bacteria. Their cell wall is made up of peptidoglycan. Okay. So they can further be divided into cyanobacteria, chemosynthetic autotrophs, heterotrophic autotrophs, actinomycetes and mycoplasma. The cyanobacteria is also known as the blue-green algae. Okay. As they are photosynthetic autotrophs. Autotrophs can produce their own food. Okay. And they are photosynthetic in nature, means they can produce their food by the help of photosynthesis. That comes under cyanobacteria or blue green algae. Okay. Examples are Anabina, nostocs, etc. Then comes the chemosynthetic autotrophs. Okay. The chemosynthetic autotrophs can, with the help of chemicals or substances like nitrates, and ammonia, they can oxidize. With the help of chemicals, they can oxidize the chemicals and they can form ATP. Okay, that is known as chemosynthetic autotrophs. Now comes the heterotrophic bacteria. What do you mean by heterotrophic bacteria? It is most abundant in nature. Most of them are saprophytes. Yes, they are useful in curd formation from milk. Okay, so in the formation of curd from milk, they are useful. In antibiotic formation, it is also used. Some are pathogenic, means they can cause diseases. Okay. Now comes the actinomycetes. These are mycelial bacteria that help decompose organic matter. Okay. They also produce antibiotics. Example is Streptomycetes. And next comes the mycoplasma, which is very important. Okay. The mycoplasma is the smallest living cell. Remember it. They are also called PPLO, pleuropneumonia-like organisms. They lack a cell wall and are non-motile. So, bacteria was having a cell wall, but in case of mycoplasma, there is no cell wall. Okay. They exhibit pleomorphism. Pleomorphism is the process by which a organism can survive without oxygen. They can survive without oxygen. They are pathogenic to plants and animals also. Clear? So, that was all about the kingdom Monera. If you have any problem, you can message me in my number. It is 79806394422. And if you want the notes, you can message me again in this number. Okay, so thank you for watching. Please subscribe if you like the video.